All right. Hi guys. I hope that you are all doing well. You can see that I just put on the board sort of the plan for the schedule for the next couple of weeks up until the next exam. So because I'm a little bit behind, we're basically going to make it through T-cell information, and the exam will just include that T-cell information, and peripheral B-cells will be later. And so we are going to have just one T-cell activation paper, and this is kind of the schedule as I will have things. Remember that we have that problem set due today and the PRR assignment due on Friday, and all of that information is posted on Moodle. I hopefully will have the paper for next week picked by Wednesday, so that I can have that for you to look at with plenty of time. And I will also either Wednesday or Friday give you a doodle poll for review session times. But that's the kind of plan for the next bit of class. And so remember that problem set is due by 5 today. You're welcome to give them to me at the end of class if you want to do so. So today, officially, our topic is T-cell trafficking. Though, as I mentioned to you, the process of trafficking that we're talking about here really is a more general process that leukocytes use. I'm putting the topic here in our discussion of the immune system in that we're thinking about naive T or B cells getting to the secondary lymphoid organ so that they can start a response. And the steps that I'm going to tell you about are key steps for that process, but it's the exact same steps that a neutrophil uses to go to a site of inflammation. It's the exact same steps that a T cell will take later in its life when it's activated in order to go to a site of infection. So we can put this process in a few different places. This just happens to be the place where I decided to put it. And if you remember, on Friday, we ended off looking at a video that showed some details of blood flow and showed a bunch of blood flowing through vessels. And I mentioned that the cells are undergoing pretty severe force, particularly shear force. And we thought about the fact that those cells are moving real fast in a real crowded area, and in some ways we thought it looked like a really crowded highway. And just like you can't just stomp on the brake and stop in the middle of a crowded highway and exit and expect everything to go awesome, the cells can't just stop in the middle of a vessel. It would really be the same kind of problem. And so just like you would do if you were driving on that really crowded highway, you have a set of steps that the cell has to do in order to get through this process. So these are the steps that we're going to see. I'm going to spend this figure talks about this as a list of 4 steps. And that is true, although we're going to focus a lot more on steps 1, 2, and 3. And then when we get to the 4th step, we're going to be like, and this happens. So when I think about this, a lot of times I think about the 3-step process. And if I say the 3-step process, I really kind of mean the first 3 steps of this. So you can see them listed here, and the first step that we're going to talk a little bit about is this step that's labeled here as rolling. And I'm going to tell you some things about rolling on the next few slides, but we can sort of think about our analogy about some sort of to predict what we might see with rolling. So if you think about that highway, you're going super fast in like some leftish lane on the highway, and suddenly your exit is coming up. What are some of the first things that you are going to do in that situation? I apologize if I can't see because I'm totally blinding myself today, standing right there. What are you going to do? Yeah. Okay. So you're going to want to start merging towards the exit. So eventually you are going to want to move to the side, right? And you're going to look and see the cell doesn't have eyes, but the cell wants to move to the side. Anything else you're going to do at this point? Yeah. Slow down a little bit. So you're probably going to slow down some, but you're not going to come to a complete stop. And you're going to sort of pull to the side, right? Really rolling accomplishes those exact same two things of helping pull the cell to the side next to the vessel and slow it down, but not bring it to a complete stop. I'm actually going to come back to this video from my recollection of this video. I think I want it in a little bit later. We can see the cells using specific types of proteins to allow rolling to occur. When we see rolling, there is this one specific kind of protein called a selectin that is involved. If you look at the word selectin, what does it have at the end? What's the end of the word selectin? And is it a word you heard earlier this semester? Yeah, Grace. Lectin. Okay, the end is lectin. And what's lectin mean? Because we saw lectin earlier this semester. What was a lectin? Yeah. It's a protein that binds to a carbohydrate. Guess what selectins are going to bind to? A carbohydrate. Shocking. So again, if we remember sort of some of this basic terminology, it helps us know what's going on. So we have these proteins called selectins that will bind to unique carbohydrate chains to help mediate this rolling process. There are, I don't know why this shows a fourth one. There are 3 selectins. They're called L-selectin, P-selectin, and E-selectin. And they are really important in those different situations I told you about. So I showed you like the naive cell moving to a lymph node, or the activated cell moving to a different situation, or the neutrophil going to the site of inflammation. They will involve a different selectin versus with L, E, or P. And we're going to talk about some specific examples later. And those selectins will bind to a specific carbohydrate. There are a few specific carbohydrates here. So you can see, for example, P-selectin binds to CD34, L-selectin binds to glycam, also sometimes called Sial-Lewis X. And that process looks a little bit like this. So here you can see Sial-Lewis X, the one I mentioned previously, binding to L-selectin. So we've got our carbohydrate, it's binding to our selectin. But what's really key about this interaction is that this is not a super strong interaction. And as a result, it's not going to stop the cell entirely. It's going to the cell is going to sort of, when it has this encounter, that's going to pull the cell over to the side of the vessel. It's going to pull the cell out of the middle of the blood vessel over to the side because of these interactions. But then as the blood keeps flowing, the cell is not going to be able to stick very well. And the cell is just going to sort of slow down and roll along. And it might catch again on another selectin. and then it might come unstuck and roll along and catch again. Often I think about the Selectin carbohydrate interaction as kind of being like Velcro, in that you can kind of be pulling it on and off really easily. So again, that's really good because it's not going to bring that cell to a complete stop and cause a traffic jam, but it is going to let the cell kind of slow down and get pulled over to the side. And so you can see this process happening here. That's this one.
Leukocytes are white blood cells that help fight infection. At sites of injury, infection, or inflammation, cytokines are released and stimulate endothelial cells that line adjacent blood vessels. The endothelial cells then express surface proteins called selectins. Selectins bind to carbohydrates displayed on the membrane of the leukocytes, causing them to stick to the walls of the blood vessels. This binding interaction is of sufficiently low affinity that the leukocytes can literally roll along the vessel walls in search for points to exit the vessel. There, they adhere tightly.
Yeah, those are later steps.
Squeeze between endothelial cells
Also later steps.
disrupting the vessel walls. Then crawl out of the blood vessel into the adjacent connective tissue. Here, leukocyte rolling is observed directly in an anesthetized manner.
But yeah, look at the cells like zipping past you can't see versus the rolling cell.
Two blood vessels are shown.
So
The upper one is an artery with blood flowing from right to left.
So yeah, notice like there are cells going so fast you can't see them versus what the rolling looks like.
Leukocytes only adhere to the surface of veins. They do not crawl out of arteries. Some leukocytes are firmly attached and are in the process of crawling.
That's the later steps.
Whereas others have already left the vessel and are seen in the surrounding connective tissue. When the blood flow is stopped temporarily by gently clamping the vessels, we can appreciate how densely both vessels are filled with red blood cells. Red blood cells do not interact with the vessel walls and move so fast under normal flow that we cannot see them. When the blood flow is restored, some of the leukocytes continue rolling, whereas all non-interacting cells are immediately washed away by the shear.
I think that's the end. It's the end. Okay. So that's kind of what this process looks like in real life. And then this is. So then I told you something. Whoa! Stop!
Welcome to this short tutorial from Pathology Makes You
No.
Black.
So I told you about rolling. We watched the rolling video. Told you about selectins, and more selectins. And so now I'm going to show you one other video that really kind of shows you this selectin carbohydrate interaction and what that looks like with rolling. I love this video. It's a really long video, and honestly, it's 8 minutes long, and it's all of cell biology. I'm only showing you two short parts of it today, but it's such a good video, except that the guy doesn't know how to pronounce the word glycosylation. But other than that.
While red blood cells are carried away at high velocity by a strong blood flow, leukocytes roll slowly on endothelial cells. P-selectins on endothelial cells interact with PSGL1, a glycoprotein on leukocyte microvilli. Leukocytes, pushed by the blood flow, adhere and roll on endothelial cells because existing interactions are broken, while new ones are formed. These interactions are possible because the extended extracellular domains of both proteins emerge from the extracellular matrix.
So anyway, that's what rolling really looks like. And it's that sort of catch, release that's happening as a result of the selectin carbohydrate interaction, depending on which specific selectin, which specific carbohydrate we're talking about. And so what I also hope that you notice back here is that there are specific interactions. So L-selectin only binds to this one specific carbohydrate. So you have to have the right combo to make this process work. And that will come up again a little bit more later. Once our cell has started rolling, it now is ready to go for the second step in this process. This second step in this process isn't sort of as easy to show in a nice little video form and things like that, but it's actually really, really important. And this step involves some signaling happening to the cell. And so I'm going to mention a little bit about the third step as well, because the two steps in some ways are tightly interlinked. Because part of that signaling is going to let the third step happen. It also does other stuff, so it's not just about the third step, but it makes an easy example to talk about the third step. So eventually, as you saw in that video, the cell is going to have to come to a complete stop, right? How do you think the cell comes to a complete stop? If we just think about this in sort of general biology, biochemistry, what is going to allow the cell to come to a complete stop? And you might compare this with the interaction we saw with the selectin carbohydrate. That was a weak interaction. Yeah, Grace?
They might tightly bind something.
There's going to be something else that's going to bind super tightly, right? And stop that cell, which is true. So there's going to be some protein that's going to be involved in stopping the cell for the super tight interaction. So right now we'll just call it the magical stopping protein, okay? Do you think this cell has the magical stopping protein on its surface ready to go all the time? So you're shaking your head no. Why no? Yeah, Josh.
It may not reach to the destination it wants to go.
Okay.
It may just stop too early because it already has.
Yeah, it might stop too early and not get to the destination where it's trying to go. Anything else? Any other reason why it probably doesn't have the magical stopping protein? Yeah, Joel. Yeah, imagine a bunch of cells all trying to stop in the same place in that vessel you saw. You would suddenly have, a clot, a stroke, a heart attack, some kind of you'd have bad news bears is what you'd have, depending on where that happened. You don't want those cells to stop by accident. You want to make sure the stopping protein is not there until we are ready to stop. Because bad things would happen if we have the stopping protein ready. So one part of the signaling that's happening here at the second step allows the cell to turn on the stopping protein so that it could do the third step. So part of this is let's turn on a stopping protein. We're also going to get some other signaling going in this cell. But you can if you decide you want to imagine it as just being about turning on the stopping protein, I'll take it. And that's listed here on this figure as activation. A lot of times I think about this as actually chemokine signaling. And so what's going to happen is there's going to be some chemokine that will bind to some chemokine receptor and lead to signal transduction in our cell. You can see a few important details here. So one thing to note is that our chemokine receptor is a G protein coupled receptor that you've heard about from Bio 250. One thing to note is that chemokine receptors can lead to a lot of changes in the cell. So there's some signal transduction stuff, but there's also some changes to the cytoskeleton, and there's also some proliferation, and there's also some adhesion. So a lot of things are happening in this cell as a result of the chemokine. Another thing, and I point this out knowing that it will come back to us later, you can also see as a G protein coupled receptor, the chemokine receptors have a specific structure, which is that it has this 7 transmembrane domains that go back and forth throughout the membrane. I mention that because we're going to see a chemokine receptor mutation in a group of patients later in the semester, and it's not gonna get the structure correct because of that mutation. So I'm pointing out the correct structure now. There are a bunch of different chemokines in the immune system. And in fact, chemokine, if you remember, is really a big long word of chemotactic cytokine, or cytokines that make cells move. But that was too long, so we smushed it together and put chemo and kine and just cut out all the middle. Chemokines have a specific naming convention, just like other things in the immune system. The way that we name the chemokines will be based on some aspects of their specific structure, but they will be named things like CXCL, or CCL, or XCL, or CX3CL. So you can see these names listed here. And so the first part is telling us something about the structure of the chemokine. Then L means it's the chemokine. It means it's the ligand. L is for ligand in this case. And then there's a number. So like one that I think about is CCL5 or CCL19. So you just get the number afterwards. And so this is kind of how we name chemokines. I will tell you, cause womp womp immunology. I will also mention at some point, IL-8. IL-8 is a chemokine. It's just, it got discovered before they figured this naming thing out. So it's annoying that it's named IL-8. It does have a name with this system, but everybody calls it IL-8. The receptors for the chemokines are also named with this same kind of nomenclature, except instead of having an L for ligand, they have an R for receptor. So sometimes I think about CCR5 or CCR7 or something like that. So if you see names that look like that, that should say in your head chemokine if it's an L, or if it's an R, chemokine receptor. And so you can see that different leukocytes will have different chemokine receptors on their surface. You can see some examples of chemokine receptors on the surface of different cells. That might be either because they're a neutrophil versus a basophil, or even because they are, say, a non-activated T cell versus an activated T cell. And each of those chemokine receptors will have a specific chemokine that it binds and responds to. As I said, the cell will receive a number of signals through those chemokine receptors to act on the cell. But the main thing I want to tell you about is the signal that's going to allow us to turn on the protein we need next. So that chemokine receptor activation is going to allow us to turn on the protein that we need for this final step. And you can see here it's listed as arrest or adhesion. Oftentimes people refer to it as either firm arrest or adhesion. I've seen it also in some textbooks as stopping. I don't care if you call it firm arrest, adhesion, or stopping. Just like the second one, you could call it chemokine stuff, or you could call it activation. As long as you know what's happening there, I don't totally care about the name. The first one's pretty much always rolling. Maybe sticking, I think I've heard, but usually it's pretty much always rolling. But rolling, chemokine activation, firm arrest, adhesion, stopping, same thing. Just like with the other two steps, this involves the interaction between two different partners. In this case, they're both generally proteins. One of them is called an integrin, and one of them is referred to as a CAM or a cell adhesion molecule. And you can see some of the CAMs listed here. And you can see examples of some integrins and CAMs listed here as well. One thing to note is that all of our integrins actually have two protein chains, some alpha chain and some beta chain. And so technically, when we name an integrin, we'll name it something like alpha 4 beta 7 integrin, because it has the integrin alpha 4 and the integrin beta 7. Some of them also have other names that they're sometimes known by, but technically we name the integrins with like names that look like this. And you can see that there are a number of other cell adhesion molecules or CAMs. And there, again, are specific interactions between a particular integrin and a particular CAM. And this is an incredibly strong interaction that really is going to stop our cell in its tracks. As we've implied in the earlier parts of the discussion, the integrins are not ready to stop the cell all the time. But like doing transcription and doing translation, that would take a long time. So it's not that the integrins aren't present. The integrins are present. They are there. They're transcribed. They're translated. They're on the surface of the cell, but they can exist in two different forms on the cell. They have an active form and an inactive form. So the integrins are on the surface of the cell in their inactive form. And when the cell gets a signal through a chemokine receptor, that makes the integrin switch. It makes it make a conformational change to go into the active form compared to the non-active form. And you can see this happening here. So here we can see at first our cell has our integrin in some weird shoe-like form that is inactive. So we've got this weird inactive form. The cell can get activated. There's some signal that goes to that integrin that makes the integrin undergo a conformational change. And now the integrin can bind to its CAM. Previously, we didn't have that binding. We didn't stop that cell in its tracks. But then after this undergoes a conformational change, you can see that happening here. These pictures at the bottom actually show more of what this really looks like. I always found this kind of neat. If you think about like a Swiss Army knife, you know how it like is bent into the thing and then you unbend it out? That's basically what the integrin is like. So the integrin, when it's unactive, is basically like this. And when it gets a signal to go into a conformational change, it goes like this. And so this is sometimes known as the jackknife conformation. So that's actually what the integrin looks like most of the time on the surface of the cell. If it gets activated, it extends out and now is able to bind to a CAM and give that hard stop to the cell. And you saw in the video earlier with the microscopy images, you saw some cells that had actually fully stopped as a part of that process. And so to give you an example, 633. Okay. This is close enough. Whoa.
Captain Marvel.
And it's Captain Marvel. Okay.
Fuses with the plasma membrane.
Okay, there. See, look, integrins. They're all bent over.
Embedded in the vesicle's membrane, fuse in the cell membrane. At sites of inflammation, chemokines secreted by endothelial cells bind to the extracellular domains of G protein-coupled membrane receptors. This binding causes a conformational change in the cytosolic portion of the receptor and the consequent activation of a subunit of the G protein. The activation of the G protein subunit triggers a cascade of protein activation, which in turn leads to the activation and clustering of integrins inside lipid rafts. A dramatic conformational change occurs in the extracellular domain of the activated integrins. This now allows for their interaction with ICAM proteins displayed at the surface of the endothelial cells. These strong interactions immobilize the rolling leukocyte at the site of inflammation. Additional signaling events cause a profound reorganization of the cytoskeleton, resulting in the spreading of one edge of the leukocyte. The leading edge of the leukocyte inserts itself between endothelial cells, and the leukocyte migrates through the blood vessel wall into the inflamed tissue. Rolling, activation, adhesion, and transendothelial migration are the 4 steps of a process called leukocyte extravasation.
Ta-da! So hopefully you see how the integrin activation works and how it ties in with everything that we've seen. And so you've now seen the rolling step that happens with selectins, then the chemokine activation step, then the arrest or adhesion step. As the video then mentioned, after that, the cell is actually going to crawl its way through an endothelial cell layer that is basically the blood vessel wall, and crawl its way out of the blood vessel wall. And the blood vessel has cells that are pretty tightly packed together, and this leukocyte is going to squeeze in between them. That process has a couple different names. It's either known as transendothelial migration, because we are going across the endothelial layer. Sometimes it's known as diapedesis. Sometimes it's known as extravasation. One thing that you will notice here is that our leukocyte is going to make some pretty massive structural changes to do that crawling. So here you can see it was nice and round in the blood vessel, and now it's all stretchy because it's trying to crawl out of the vessel wall. And there's a lot of kind of cell biology that goes on here. There's actually a lot that we really don't have great answers for of how this works. In all the other steps, I've told you specific protein-protein interactions that are important. Here, I'm just going to say, we think there's some chemokines involved, but we don't really have great answers of what's important. So I'm just kind of going to say, and then this step happens because I can't give you as much mechanism on this step as I can with the other steps. There is an immunologist named Pedro Valencia who draws immunology-related comics, and they're really fantastic. And this was his comic last Monday was actually on extravasation. So if you don't understand that process that we just saw, here is a comic. You got this nice, tight wall of the blood vessel, all the cells coming together, and then you have this little leukocyte that wants to come through and basically squeezes its way out of the blood vessel to go into the tissue below it. So you can go to Pedromics and see all his fabulous cartoons. So that's kind of this big picture process. And all of this process, all of these steps that you have seen will help a white blood cell leave a lymph node or leave a blood vessel to go to a place. But we can also think a little bit about ways that this can sort of be unique for different situations. So if I like this analogy, but also this analogy is rapidly becoming like old-fashioned. So I apologize if I tell you and you're like, I don't know what you're talking about. So imagine that I want to make a phone call to someone else in the United States. And how does my phone know, like, it should connect to a person in New Jersey as opposed to North Carolina, as opposed to California or something like that? How does my phone know what to do? Like I said, this is getting very old-timey. Yeah, Andrew?
Doesn't each phone number has like the area code?
Yeah. So remember, the first 3 numbers of your phone number are an area code. So like, if I dial 973, that means it's New Jersey. And if I dialed 919, it means it's North Carolina. Basically, this process that we just saw with the 3 steps, you can kind of think of as being akin to that area code. Because what you can imagine is every leukocyte is doing this. But every leukocyte doesn't necessarily have a way to be like, to know it's a neutrophil and that it needs to go to my ankle to help my ankle be swollen. I'd like it to not go to my ankle. Don't go there. Or it's a T cell that wants to go to a lymphocyte or to a lung to get rid of SARS-CoV-2. They're all using the same 3-step process, but the way that they're doing unique things is that in each step, there's an important pairing of the 2 different proteins. And so one particular cell type is only going to go to the right to a certain location if the correct Selectin carbohydrate pair is there on the white blood cell and on the vessel. And then if the right chemokine and chemokine receptor are there, and then if the right integrin and CAM. And so again, it's 3 different steps where you have to have just the right thing that indicate, yes, this is the lung. Yes, this is the skin. Yes, this is the GI tract. To mark different locations for different cells. And so, exactly which integrin and which selectin and which chemokine and chemokine receptor become very important in indicating kind of the address labels around the body to help cells know exactly where they are supposed to go. And so I want to show you this in terms of kind of some specific ideas of what this looks like in each of these 3 situations. But really what I'm showing you is it's going to be the same steps over and over again. It's just that we're going to see how they would look slightly differently as like address labels for different scenarios. And so the first scenario we're going to think about is the scenario of a neutrophil that wants to go to a site of inflammation. So this is going to be I'll say my weird question later. So we can think about exactly which molecules we see in this case. And so if we have a neutrophil going to a site of inflammation, we have a step with selectins. We have a step with chemokines. We have a step with integrins. And I can't reach that high. In the case of this situation, the neutrophil has the carbohydrate And that carbohydrate is PSGL1. And the site of inflammation has the selectins, which are actually both P and E selectin. This is my weird question I have for you. Is a neutrophil ever like not stop being a neutrophil, or is it a neutrophil its whole life? Is a neutrophil suddenly like a cell of the eye? No, it's just a neutrophil, like its whole life. Its whole life, it wants to go to sites of inflammation. Its whole life, it would love to go do this process. As a result, it pretty much has PSGL1 on its surface its whole life. It's like, here, I would like to come to a site of inflammation. Here's my thing to start me. It's just, because it's a neutrophil, it just always has that on its surface, ready to go to a site of inflammation. Is a site of inflammation always a site of inflammation? No. Like this spot of my skin is fine, right? This spot of my skin does not currently want neutrophils. So this spot of my skin does not currently have any P or E selectin to try to sort of bring neutrophils in. We're going to induce, if something happens and I get some inflammation there, that part of my skin is going to be like, oh, I got to bring in some neutrophils, and it's going to start expressing P and E selectin. What you'll notice throughout this process is that the selectin is always the one that's turned on and off, and the carbohydrate is always there permanently throughout this whole process. Then we're going to have a chemokine, and the chemokine in this case is IL-8. The chemokine is also inducible. So the chemokine is, this happy good part of my skin is not making any IL-8 right now. It's not trying to get neutrophils to come there. Whereas down there on my ankle, there's probably lots of IL-8. It's probably doing a lot to get neutrophils. And in fact, that we are really seeing that neutrophil have what I will just call the IL-8 receptor. I believe it's CXCR8. And it has that all the time. It's like, I'm ready to come to a site of inflammation. Site of inflammation, you please tell me. We're ready to come. This receptor will then turn on the integrin, which the integrin in this case is LFA1, which binds to ICAM, which is on the next slide. So whichever partner has the chemokine receptor is also the partner that has the integrin. And I believe this is alpha 4 beta 1. I can check. Was I right? Let's see. Oh, no, I was wrong. Sad. Alpha L beta 2. That's why I don't ever call it that. And so it's the same three set of same set of 3 steps we saw before, but because we're now thinking about the specific situation of a neutrophil going to specifically a site of inflammation, we can see specific versions of this, or cells that are going to be part of this. Oops. We can also imagine the same situation if we are thinking about an adaptive immune cell, like a T cell that is going to some kind of site. And when we think about a T cell, which is really the point of doing this in this part of the semester, is that we're doing this to help us think about T cells, there are 2 places that T cells might want to go. One of those places is what happens to this T cell when it's shown here in red, which is the T cell that has never found its antigen. It came out of the thymus and it's never found its antigen. It's just going to spend its time looking for its antigen. Please, hoping it finds its antigen. And it's going to go to secondary lymphoid organs to do that. And so you can see this red T cell is going to leave the blood vessel and come into the lymph node. And it is going to then look around the lymph nodes. And so it's really going to be sort of doing this secondary lymphoid organ thing. This is a little bit of a point that is going to come back later, but I'll just tell you a little bit now. It's not going to do its function that first time it sees antigen in the lymph node. So sometimes people get really worried about a killer T cell killing the lymph node. Oh my gosh, I got a vaccine and my killer T cells are going to get activated for SARS-CoV-2 and they're going to kill my lymph node. It doesn't kill the first time. It just gets activated. It's going around the lymph node. But once that cell gets activated, see here you can see, oh my gosh, it found its antigen. It got activated. It changes. So now it's blue instead of red. And now that cell does not want to go to lymph nodes anymore. Now that cell wants to go out to some other body site and find the infection and kill it there. And so that cell is going to change where it's going. So a naive cell wants to go to secondary lymphoid organs, like the lymph node, while an activated cell wants to go to other sites of inflammation. And these 2 scenarios will kind of give us our other 2 examples we can see for trafficking. So we can see either a naive T cell that wants to go to a secondary lymphoid organ, or we can see an activated T cell that wants to go to a site of infection. I'm going to talk through the details of that naive T cell going to the secondary lymphoid organ first, and then I'll mention some details about, we'll sort of do a compare and contrast with the activated T cell going to the site of infection. This gets its own slide because I was so confused about it for a long time as an undergraduate. So when we look at lymph nodes, we can see blood vessels coming in and out. And I've shown you those blood vessels before. Sometimes they are listed as HEVs. The HEV is the specialized vessel that's in the lymph node, and it's the specialized vessel that those T cells are trying to get out of. And so you can kind of think of it as the special vessel that is bringing T cells into the lymph node. HEV stands for high endothelial venule, which is listed on the top here. And your textbook just suddenly starts calling it an HEV and doesn't really tell you what an HEV is, other than just being see, it goes into the HEV. So HEV is a high endothelial venule. It's basically a specialized blood vessel in the lymph node that's really good at allowing naive T cells and B cells to come into the lymph nodes. And took me an embarrassingly long time to understand why the heck it was called a high endothelial venule. The reason is because see these cells that make up the wall, which are the endothelial cells? They're tall. They're like tall, skinny rectangles. So they're high because they're tall. So it's a venule that's made of high endothelial cells. And like I said, it took me an embarrassingly long amount of time to have any clue what the heck that meant. So I'm just telling you, make your life easier. Does a high endothelial venule cell, does a cell in the HEV, ever not want T cells? Particularly naive T cells? No. It's like always like, T cells, come here. And so again, it's going to have some products that it is making all the time. It's always going to make a particular carbohydrate. Which is called sialylus X. You can see it here too. They're calling it CD34. Whatever. We'll call it either one. I don't even care. So basically, HEVs always have that carbohydrate. And you can see here's the HEV always having that carbohydrate on its surface. Because that HEV is always trying to call in naive T cells. The naive T cell, when it's a naive T cell, when it wants to go to secondary lymphoid organs, is going to have L-selectin, which binds to this carbohydrate. So that's going to allow that naive T cell to start the process of going to the lymph node. As a little bit of a spoiler, do you think the activated T cell will have L-selectin? No, because the activated T cell does not want to go to the lymph node anymore. So it's going to stop making L-selectin, and it's so that it can sort of traffic somewhere else. It can get a different area code to go on. So it's going to stop its L-selectin production. The HEV is also always making chemokines, specifically CCL19 and CCL21. When the T cell is naive and when it would like to go to the lymph node, it will have the chemokine receptor, CCR7, on its surface. So we've got this chemokine coming from the HEV, binding to the chemokine receptor on this T cell. When that T cell is activated, do you think it is going to have CCR7 on its surface? No, because it doesn't want to go to the lymph node anymore. It wants to go somewhere else. So it's going to have something else going on in that chemokine step. It's going to stop making CCR7. The lymph node is always making CCL19 and 21 because it's always like, T cells, naive T cells, come here. And then you can see that we have LFA1 again as our integrin binding to ICAM1. So in this place, it's actually the same proteins. And so you can imagine if I wanted to call somebody in New Jersey, it's 973. And if I wanted to call somebody in North Carolina, it's 919. So we both start with 9. But because the rest of the combos are different, it leads to 2 different area codes. So yeah, here we have the same proteins, but because we've paired it with other things, it becomes a different area code. And then when we look, and I'm not going to give you specific examples for the activated T cell in the site of infection. And the reason why I'm not going to do that is because to have an area code, you need to have an area code for a specific place. And so the area code for an activated T cell to go to the intestine is different than the area code for an activated T cell to go to the skin, which is different than for that activated T cell to go to the liver, which is different than if it goes to the lung. But what you should know is that it is no L-selectin, no CCR7, because it does not want to end up back in a lymph node and maybe kill the lymph node. It now wants to go where the infection is and do something there. And the specifics of what it does have will vary based on exactly which body location it's going to go to. And I'm not just going to like sit here and read you, here are the chemokines for every body location, because that would be really boring. And because we don't know them all. And so all of that is going to allow our T cells to get into the secondary lymphoid organ. One other thing that you should notice here is that in our secondary lymphoid organ, it's not just a sort of homogeneous location. There are unique individual sites within each secondary lymphoid organ. So here you're seeing a lymph node at the top. At the bottom, we've got the lymph node versus the spleen. And what you can notice is that the spleen has some other stuff, but areas that also look like the lymph node. So I kind of just focus on those areas and say that we're talking about basically the same thing. But what you can notice is that there are B cell zones, and there are T cell zones, and there are all sorts of different zones. So once our cell leaves the HEV, once our cell leaves that vessel, it then is going to have to travel around the lymph node to go to its correct location. And that is also happening due to chemokine gradients. So the entire lymph node has sort of different levels of chemokines in different locations that will attract different cells to different places. So there's a lot more going on here with chemokines than just in that early step, but you should just be aware of that. One other thing I just wanted to point out is that as we are learning much more about the immune system, we're learning about things that maybe we originally thought were simple, and we're like, oh, wait, that's a thing? So, for example, we've actually realized that when you raise your body temperature, most of these trafficking proteins work better. So fever not only does things like block microbial replication, it also helps integrins work better and helps trafficking work better so that you can have this process working better when you are sick, which makes sense. And so there are a number of other kinds of benefits that might come up from some of these things that we've seen and thought about in the past. All right, so all of this discussion has really been, as I mentioned, kind of in service of us thinking about the timing of adaptive immune responses as we look at the timing of T cell responses. And so what you will notice is the first thing that has to happen is those T cells have to get to the secondary lymphoid organ. We also have to get antigen to the secondary lymphoid organ, which I mentioned a little bit about before. I'll mention some more in an upcoming lecture. And so we've just talked about all the things that make that take a little while. It might take up to a day or so to get everybody into the secondary lymphoid organ before we can then start the process of activating our T cells. This is showing us a little bit of information about T cell activation. We're going to be going through more specific details that we see here in the next couple of days, but I want to kind of hit some highlights so you know some things to be looking for here. So first, you'll notice that our T cell, whether it's a CD4 cell, which is at the top, or a CD8 cell, which is at the bottom, needs to interact with a cell presenting peptide on the correct type of MHC. So MHC class 2 up here, MHC class 1 down here. And we're going to have some protein-protein interactions that allow that T cell to become activated. When that T cell becomes activated, it's going to make some new products, which we'll see more about. It's going to undergo some rounds of division. So you can see now we have more of the T cell. And that cell is also going to undergo a process of differentiation. So that cell is now going to gain new functions. It's going to sort of grow up and be able to do a function that it could not do before. So it needs really ideal events to happen at this first part of activation so that that cell can be told to divide or proliferate, and so that cell can pick up its function. I've told you about CD8 T cells having the ability to kill or be cytotoxic T cells. Those CD8 T cells, when they are naive, are not able to kill, but after they've gone through this activation process and been differentiated, they will become those cytotoxic CD8 T cells. The CD4 cells just start out as a naive CD4 cell. After activation, they become those helper T cells that are producing a whole lot of cytokine. Much of what we're going to see to make that activation happen will include signaling from the T cell receptor. And on Wednesday, we are going to be in the weeds of signal transduction in the T cell receptor. So be ready for like serious signal transduction. This is, your textbook tries to make the whole thing like one figure. And we're going to just go through this all in like a whole bunch of detail of the exact details of what happens with lymphocyte receptors. And one thing I will just tell you is that there are these general themes of what kind of proteins participate in this process. And we're going to see them in detail with the T cell. But it's actually the same kind of theme with B cells too. So I just picked to tell you about it with T cells instead of with B cells. There are a couple other things about turning on T cells that are really important. I'm going to switch the order of the next couple of things. I mentioned to you before that the T cell receptor has rather low affinity for MHC plus peptide, and that we need some additional proteins to help kind of stabilize that interaction. And that was a key role of CD4 and CD8 to help bind the cell to the MHC molecule. In reality, there are actually a ton of proteins that are involved in this process of holding the two cells together and stabilizing them, because they actually need to be in contact for a while in order to get all this signaling to work out. And people have actually like done timing experiments of how long they need to be together for like hours. It turns out the way that happens is that T cells just use more integrins. They just use integrins to hold on to other cells as well. And so in reality, our T cell has a whole bunch of proteins, like the T cell receptor, like CD4, etc., and also a bunch of integrins, really holding the whole thing together. And those integrins almost make a ring around the T cell receptor. So it's sort of like there's this little ring of signaling, which is the stuff we're going to see on Wednesday, with a wall around it, holding the two cells together. And it actually ends up working pretty nicely. So here are my two cells. Oops. Here is some MHC class 1 with peptide. Here is T cell receptor, right? I'm going to have this great big integrin wall on either side holding the cells together. And if those cells if this cell, say, wants to communicate across this area, if it wants to secrete things, it can secrete stuff in this little area, and the stuff can't diffuse away because there's a wall around it. There's this whole like cylinder of just sort of protected area. And so we're going to actually see a bunch of what's known as directional secretion, where the cells can actually sort of talk to each other inside this integrin wall. To the point where, I mean, we can actually like see these proteins make like rings on the surface. It looks like a Target logo if you look at where different cells or different proteins are set up on the surface of these cells, which is known as the immunological synapse. The one last thing I just want to mention is that in order to turn on a T cell really well, the key for turning on a T cell, and you will see so much more about this, the T cell usually needs some other kinds of signals. And so, in particular, it needs this thing called signal 2 or co-stimulatory interaction. Oftentimes, we're going to need some cytokines too. And so we're going to see some of those cytokines, and we're going to see that co-stimulatory interaction. That co-stimulatory interaction is not there, everything falls apart. Nothing works without the co-stim. I mention that because when you're thinking about the PRR assignment for Friday, it does bring up the co-stim interaction. That's actually kind of a key thing in those readings. And so, we're probably not going to get to it until Friday, although we'll see how I arrange the slides on Wednesday, but know that co-stim interaction is required for that T cell to be activated. And that's a key thing that you're going to see in those readings. Okay? So make sure you turn in your problem sets by 5. And I will see you guys on Wednesday.